Before I start my formal talk, I have two of my kids in the audience, and I wanted to start with a lesson. Last year, I was quite honored to receive a medal from President Obama. But what I want my kids to know is 10 years earlier, for the same thing that I got a medal from the president for, I was fired. So it really shows you what difference 10 years make. So I guess I'm going to tell you for the rest of the talk what I got fired for. I'm going to start a few summers ago. A number of you are from Europe. A few summers ago, there was an outbreak, an E. coli outbreak in Germany, but it was different than anything that anybody had ever experienced before. This outbreak sent over 700 people to the intensive care unit, and unfortunately sent over 50 of the people to the morgue. And we were lucky enough to be involved in understanding this outbreak. A researcher in Germany, using a machine that we had developed, using a semiconductor chip for the first time, was able to decode the genome of a superbug, or a Frankenbug, that had caused these 50 people to die. To give you some perspective, the genome we decoded was 5 million letters long. I want to give you a heads up that I'm from the biological or life sciences. We don't have any new math or new physics, so we try to keep most of you out of our field by our vocabulary. If you get rid of the fancy vocabulary, what we do is pretty simple. So we decoded 5 million letters, these ACGT, that specified this microbe. But I wanted to give you some perspective. A decade or so before that, I had received a PhD, and over a 6-year period, I decoded 9,000 letters, which represented one gene that was involved in the development of the nervous system. So how did we get here? How did we go from being able to get a PhD and take 6 years to decode 9,000 or 10,000 letters of a genetic code, to being able to decode on a disposable chip 5 million letters in 2 hours for $99, put that information on the internet, have scientists around the world look at the genetics of this microorganism, quickly make probes that were able to be used to screen plant material or food sources for it, and understand why this microbe was resistant to 14 strains of antibiotic. How did we get here? Well, my journey starts with a personal experience. It starts with my son, Noah. I think I stole the punchline. Noah is okay, but he was rushed to the newborn intensive care when he was born with difficulty breathing. And I wanted to understand at that moment, why wasn't my son well? What was special about my son? Now, in 1999, something else had happened. The public effort and a private effort by Craig Venter had decoded the first maps of what it meant to be human. Basically, they took either 50 people and put them together and decoded the 3 billion letters that make up a human, or in the Craig Venter case, 6 people, about 70% Craig Venter, and decoded what it meant, to be human. These were maps, consensus maps, what we have in common as humans. Now, I sat there realizing that I didn't have 10 years and $3 billion to decode Noah's genome. I wanted to know at that moment, not what Noah had in common with humanity. I didn't want a generic map of all of mankind. I wanted to know why my son Noah was having difficulty breathing. What was different about his genome? Not in common. And that night, while waiting in the intensive care unit, I saw a computer magazine with a Pentium chip on it, and they mentioned that the new Pentium chip at the time had 10 million transistors. And I realized that my entire industry had been going about the problem in the wrong way. To decode genomes, larger and larger genomes, we had assembled teams and automated those teams in the same way that Henry Ford built cars. And I realized that the solution was not in more automation and more people and bigger facilities, but to miniaturize the process. Now, people had tried to miniaturize the process. I had gone to a decade's worth of talks where people were taking the gold standard at the time, something known as Sanger sequencing, the technique I used as a graduate student, and tried to miniaturize it. But it didn't work. And I felt miniaturizing Sanger sequencing would have been like Noyce and Jack Kilby taking vacuum tubes and trying to put them on a single substrate versus a transistor. And so what I knew of is I knew of an inferior technology that had never been used to sequence, but had been used to see or used to detect a single change in DNA, a single base. And I figured if I could put this on a single substrate, again, taking a book right from Noyce and Kilby, that I could do this in a parallel way. So when I did my thesis, I used about 1 square foot to decode about 12 strands of DNA. By putting it on a single substrate, I could decode in the area that would be on the end of a single human hair about 4 strands of DNA. And we were able to immediately have 1,000 to a 10,000-fold improvement over anything that had come before. Now, it turns out I'm not going to go into the details of the chemistry of the DNA, but you end up spending a lot of your time preparing your sample from a person, from a bacteria, from any organism. And then you sequence it, you decode the letters. So we worked out 2 breakthroughs. We reduced the getting little pieces of DNA into a fast process, and then we did it all on a single substrate. One of the great things of having a tool that no one else has is you're able to go around the world, find the best scientists in the world, and they can do amazing things with your tool. Again, at that moment in history, we could decode DNA 1,000 to 10,000 times faster and 1,000 to 10,000 times cheaper than anybody else in the world. And in fact, the company I started, 454 Life Sciences, had a 2-year monopoly on high-speed, or what became known as next-generation DNA sequencing. And we were able to do genomes. We were able to learn new biology, how our genomes were regulated. We were able to sample people's stomachs and find out they had whole ecosystems in them, and decode ancient DNA. I'm going to spend a few minutes talking about some of these projects. First project we did with a pharmaceutical company. Pharmaceuticals are developed literally by finding a key that fits in a lock. The lock is the target for your drug. And that's how they were developed for decades. Well, J&J had a key that they knew could kill tuberculosis, but they couldn't improve it because they didn't have the lock. So for the first time in history, we found them the lock simply by reading off the 5 million letters that were in the genomes that were sensitive to the drug, and 5 million letters from genomes that weren't sensitive to the drug, and the only difference was a mutation in the lock. So they had their lock, and they were able to develop their drug. Got a call from, actually called a researcher at Harvard. He had published that people were sensitive or not sensitive, or responsive or not responsive to one of the newest cancer drugs. And that cancer drug had just been pulled off the market because of low efficacy. They weren't giving the right drug to the right person. He said, let's sequence people's cancers and make sure we give them the right drug by having the drug only given to people that have a mutation that are drug targets. And so for the first time, we were able to sequence, instead of a whole organism, as I talked to you in the tuberculosis case, the same gene in your genome, but hundreds of thousands of times, because each cancer cell is different. Cancer is a disease of your genome. And so by sequencing the same gene, which was the target of this drug, hundreds of thousands and then millions of times, we were able to show a patient had 1 or 2% of their cancer cells resistant to this drug. And we knew those cancer cells, if you were treated with this drug, would be the ones that grew and eventually killed the patient. And Matthew Meyerson, who was the physician scientist we worked on after we did the sequencing, changed the therapy, and the patient responded. I thought that was wonderful, but our lawyers called us and said we had to mark each of our machines and each of our kits not for human use because it hadn't been approved by the FDA. The good news, though, is that physician scientists, for the next 8 years after we did that publication, adopted this technique widely, and every center, every major medical center around the world, if you have a cancer, will sequence either a few genes or the whole genome of that cancer to make sure you get the right drug. So we were able to have a huge influence around the world on cancer therapy, and now it is FDA approved. We had another case where one of our collaborators had 3 patients that all died after receiving a transplant. When we sequenced tissue from the people that died, we found a virus that was killing them. But normally this virus doesn't hurt you. But when you get an organ transplant, you're immunosuppressed. And in that case, this virus, which is normally non-pathogenic, becomes lethal. Important lesson here is it's both your genetics, whether it's your own genetics or that infection or virus, as well as the environment. So if you're not under immune suppressant, it would have been fine. A fun thing about this is they featured our machine on the set of the movie Contagion. Unfortunately, the movie was not a big success. Now, it turns out that you can use DNA literally to heal, feed, fuel, and secure the world because it's universal. Big mystery, honeybees were dying across the United States. Researchers sent us just material from those hives that had died. We extracted the DNA, and sure enough, we found a virus in the hives that were killing the honeybee. And if you took that virus and infected a hive, it would collapse. Now, before we say we found out what's killing the honeybee, I want you to remember those transplant patients. Something in the environment is making these hives sensitive to a virus which otherwise wouldn't be lethal. So I like finishing projects. We had started this technology because my son wasn't breathing and I needed a way to analyze his genome. My son is fine. He's in the audience today. So we had to sequence somebody's genome. So we chose Jim Watson, who was the person, along with Francis Crick, who discovered the structure of DNA and gave us the basis of heredity. Now, that's pretty good. So there's good news there. Science Magazine Breakthrough of the Year. I felt pretty good. The bad news, as I already mentioned, is I was fired. I was fired because the board figured there really wasn't much use for this whole genome sequencing stuff. Hadn't it been done? So our 2-year monopoly didn't end well for me. But as I said, 10 years later, I got a medal for it, so it was okay. Well, anyhow, when you're out of work and you have mouths to feed, you figure you want to do something. And most of us know what we're good at. I was good at DNA sequencing. But again, when I was first, I had a 2-year monopoly. We had 100 million sales in literally our first year because there was nothing else out there. But after I got fired, other people came, literally became an industry. Many other companies, many in California, came and brought machines to the market. And I wanted to have an impact on sequencing. But in this case, I wouldn't be first. So I needed another strategy. And what I wanted to do was not take a page from Noyce and Kilby, not take the idea of the monolithic idea to sequencing, but I wanted to take your entire supply chain, the $3 trillion that you have put into this supply chain over the last 50 years, and I wanted to leverage it. And I knew semiconductors win. Whether it was for computing or for cameras, they win. I remember seeing on my iPhone the news that Kodak was restructuring and going into bankruptcy. And I remember when the first digital cameras came out, how people said they would never be as good as film. So what I wanted to do was make a chip that would read biological information. Now, usually I hold up a cell phone, I show people the CMOS imager, and I say, look, everybody in this room is carrying a device that converts photons to electrons and then gets beautiful pictures. I wanted to do the exact same thing, build a chip, but instead of seeing photons, I wanted to see ions. So I put together a team. Starting in 1993, I had been interested in something called an ISFET. Most of you are electrical engineers. When I speak to a group of chemists or molecular biologists, I ask them to raise their hand if they've ever used a pH meter, and all the hands go up. I'm not sure how that would go over here. But every modern pH meter has an ISFET that detects that pH change. As a chemical engineer, I know when I make a strand of DNA that every time I add a base, a hydrogen ion is released. And I knew that all DNA sequencing is based on this synthesis. You synthesize DNA from a template by knowing what's added. If an A is added, you know there was a T in the opposite strand. If a G is added, you know there's a C, and so forth. That's how all the DNA sequencing basically had worked. So I wanted to sequence now, instead of on gels like Sanger, or instead of with Firefly enzymes like we did at 454, I wanted to directly sequence on a semiconductor. I brought together an amazing team, a number of researchers in the audience today, and I said, let's make a chip that would use ISFETs in a massively parallel way. Let's put structures on top that will hold DNA. Let's put little micro machines, in this case enzymes, and let's sequence on a chip. And instead of sequencing 4 strands of DNA, like we did at 454 on the end of a human hair, we would do 400. So that would give us a 100-fold increase. Now, before I built this, I also, since I was naive, called around and found all the reasons why people told me it couldn't be done. And I just hired people who said it could. So we did it. Again, it had been an idea that had been talked about for decades, but nobody had pulled it off. And we were able to pull it off by hiring what I call people that don't know better, putting them in a room and telling them to do it. So we were able to make a semiconductor device. We were able to microfabricate chambers on top. We were able to do the surface chemistry and the biochemistry that would copy strands of DNA. But again, this time, millions in parallel. And we were able to have a machine that instead of costing $500,000, which is what the other DNA sequencing next-generation machines cost, because they were built literally in your own facilities with optics, they were giant microscopes, we would use the same factories that made Xboxes and the same foundries that made all of your chips. And that's how we were able to decode a bacterial genome for $99 on a disposable chip, instead of taking years or millions of dollars as it had cost previously. Now, if you recall from an earlier slide, when we first announced that we had a breakthrough in sequencing, we had 2 types of people: the people that liked it and the people that told me it wouldn't scale. The people that liked it, I hired as advisors to my company. The ones that didn't like it, I avoided. Well, this time, I was a little bit gun-shy. We were going back to publish in Nature. So this time, I didn't want anybody writing to me that it would never scale. It would never go from viruses to bacteria to human. So what we decided to do was sequence the genome of Gordon Moore. And then nobody could argue that it wouldn't scale. And that's precisely what we did. Got my wife pretty mad, but we did sequence the sequence of Gordon Moore. She says, why do you always sequence 80-year-old plus men that aren't important to the healthcare system? Anyhow, she's a doctor. So we sequenced Gordon Moore, and we showed for the first time that you could actually sequence on a path towards this $1,000 genome. It's not going to affect you every day if it costs $1 billion to sequence a genome. But right now, each of you costs the healthcare system $6,000 a year. So it's not a bad cost, $1,000 incremental, to guide your health potentially for the rest of your life. And Gordon Moore really was the first of the rest of us. I have Keith Fife in the audience because we showed we could sequence a bacteria for $1,000. And I wanted to do a human for $1,000. And so what we did is we moved to a newer node. Instead of sequencing 400 pieces of DNA on the end of a human hair, we would sequence 14,000. Some beautiful architecture, which actually has been published at this conference. And now, instead of sequencing bacteria in that 2 hours, you can sequence any of you on a chip. One of the things that happens very often when I give talks, or even listen to commentary, is people talk about biology and DNA sequencing going faster than Moore's law. And what I want to show you with this chart today is it's an illusion. We're not going faster than Moore's law, we're simply catching up. If you look at that first next-gen sequencing, it was at a density that was equivalent to the density that you guys were using in the 1980s. If you look at that chip I showed you that did the bacterial genome, it was from a node from the 1990s. And if you look at anything you guys have done on those nodes, you have this 1,000-fold improvement. And so we've gotten these twice 1,000-fold improvements or 1,000,000-fold improvements just by going to higher and higher densities. And in our case, because we're on semiconductors, literally just going to newer foundries. This is important because it clearly says once we catch up to whatever node we can use for the analog front of what we're doing, we then have to wait for the rest of Moore's law. And in fact, we've run into this wall now, not at the density that we can sequence, but at our ability to analyze the sequence and analyze that information. So while you saw this super Moore's law over the last decade, it's just that we're catching up. And once we're at this $1,000 genome, which is where we are now, we really are going to proceed more along a pace that all of us are proceeding. I do want to show you just some. Did I lose my? No. Do I have good sound? Good. I do want to show you some fun projects. So we had sequenced Jim Watson, we had sequenced Gordon Moore, and I do. Okay, are you on the lectern? Okay, we had sequenced Gordon Moore, we had sequenced Jim Watson. My nephews were at Burning Man, and we actually sequenced Charles Townes, the person that had given us the laser, and we beamed his genome into outer space, just to show some of the cool things you could do with genome sequencing. Now, I also mentioned that my first machine was on the set of a movie, but what I didn't say is that Noah said, well, that movie wasn't a big success. So the good news also was my next machine, the Ion Proton, which can do these $1,000 genomes, was on the set of Jurassic World and on their website. And the good news is Jurassic World was the biggest box office hit opening weekend. So I was kind of redeemed there also. Now, while we were doing serious science with our technology, we decoded Neanderthal. really understanding why we're different than Neanderthal, allowing us to identify genes that are evolved in higher cognitive function. While other people used our machines to sequence mammoth, I was always a little chagrined that when we had a Nature or Science publication, I would read either in the Wall Street Journal or New York Times something completely different. And so when we did the Neanderthal genome, I was looking for a great story that we would uncover what makes us special. Neanderthal never crossed a body of water that he didn't see the other side. When we came out of Africa, we were on Easter Island. Clearly something different. But instead of talking about it, it said how scientists were going to use the differences to recreate Neanderthal. And sure enough, you know, that's what they were doing with sequencing in Jurassic World. So that was just a little fun. All right, I want to change gears a little bit, because I was successful because I found the smartest people in the world that could do great things with semiconductors in, as I learned today, a three-dimensional way. We would put things on top of the semiconductors, and then we would put that into context. It doesn't help to decode a bacteria that's killing people unless you can put that information into the cloud, other people can understand it. And so what we wanted to do is we wanted to generalize that paradigm and realize that we were at an amazing inflection point in medicine because of high-throughput DNA sequencing, and an inflection point on the computer side. So what we wanted to do is say, can we generalize this paradigm? Can we make new devices, put that information in the cloud, but instead of just having experts analyze it, have experts in machine analyze it and create a virtuous circle? And so what we did is we created an incubator, and we started 4 companies, all with this exact same strategy. They would need devices. I'm recruiting all of you, jonathan.rothberg@gmail.com. They would put information in the cloud, and they would understand it. But again, we would put something special on top of those devices. The first of these 4 startups we call Butterfly. And Butterfly is building, and has built, successfully built, an entire ultrasound machine on a chip. So instead of having a cart that costs $100,000 and $60,000 worth of probes, piezoelectric probes, we would now have a low-cost integrated circuit, and we would put MEMS on top. So we've done that. But if you make a low-cost ultrasound machine, that's only part of the problem. There are only 30 radiologists in Uganda, for example. So what we've done is partner it with the cloud and this deep learning. And now our software can guide any of you to acquire the correct image, whether it's cardiac or a fetus, and analyze it. And again, create the circle. So if a cardiologist is using this technology and he corrects the machine, it gets better and better. And so we've created these chips. We'll be launching product this year, and it will allow anybody, any of those 40 million healthcare practitioners, to collect information on ultrasound and have it analyzed in a useful way. And in the same way that we democratize DNA sequencing, our goal now is to democratize medical imaging. I did mention that I was fired for the same thing I got an award for, but I want to make clear that all of our success comes from getting the best people in the world, whether they're advisors or the team that you bring together. And I also want to make clear for those of you who are academic that when you come to a company, you can do great publications and advance your career. Nevada Sanchez, the co-founder of Butterfly, was one of Forbes 30 under 30. Also, we don't operate in a vacuum. The best advisors in the world work with us. So David Ferrucci, who gave us Watson and Jeopardy, Watson that beat Jeopardy, is an advisor to us and helping us on the machine learning. And Max Tegmark, who's a physicist, is helping us on the imaging. I want to thank everybody. Again, list is too long to list for all of you in the audience who have helped me, and look forward making devices that can change the lives of people we love. Thank you.